So now we are going to read about the isolation of the viruses, which comes under the diagnostic virology. So isolation of viruses are mainly done for the research purposes, not for the diagnostic purposes. But we have to discuss it under the diagnostic virology. And as I have written here, the isolation of viruses are not performed routinely from diagnostic virology. Rather, it is basically done for the research purposes, for seeing the growth of the virus, for seeing the characteristics of the virus. So this isolation of viruses may be asked in as a short note or a long question in the university exams. So there are various ways by which we can isolate the virus, that is the animal inoculation, egg inoculation, or the cell line cultures. Now we haven't mentioned the animal inoculation because it is outdated now and it is not used in these days, as some controversies arises due to use of those animals for the experiments. So those animal inoculations have been removed or not from nowadays. So in today's world, the most commonly used methods for virus isolation egg inoculation are the egg inoculation and the cell line culture. So in egg inoculation, we take the embryonated hen's egg, and in the embryonated hen's egg, we inoculate the virus at four different sites. So there are four different sites for us to inoculate the virus, where virus can grow. The first site is the yolk sac inoculation. So the yolk sac of the egg can be inoculated, and the yolk sac is used for inoculation of the arboviruses, the rickettsia, chlamydia, and the Haemophilus ducreyi. How can you remember this? So this can be remembered as rich arboviruses are present in yolk sac. So rich arboviruses are present in yolk sac. Rich arbovirus means rich. Ri means rickettsia, C means chlamydia, H for Haemophilus ducreyi, and arboviruses means arbovirus. They are present in yolk sac. So rich arboviruses are present in yolk sac. The next site for inoculation of the viruses is the amniotic sac, where we grow the influenza viruses. So amniotic sac is used for the influenza viruses. How can you remember this? See here. Here in the influenza, there is in, and here ni. Okay, can you see here ni? So in the amniotic sac, we grow the influenza virus, or amniotic sac is used for the inoculation of the influenza virus. By that you can remember. Next comes the allentoic sac. So allentoic sac, since it is a larger sac, so it is not used for just the growth of the viruses. Rather, it is more commonly used for the viral vaccine development. And the two most commonly developed vaccines in the allentoic sac are the influenza vaccine and the yellow fever 17D vaccine. Okay, influenza vaccine and the yellow fever 17D vaccine are used developed in the allentoic sac. And why we choose the allentoic sac? Because the allentoic sac is a larger sac in the embryonated egg. Therefore it is easy, therefore we get the vaccine in a sufficient amount, and hence we grow it in the allentoic sac. The next site is the chorioallantoic membrane. The next site is chorioallantoic membrane, and this chorioallantoic membrane is most commonly used for the pox viruses, for the vaccinia and the variola strains. Okay, and there we see certain characteristic features that we will see later on. Next we come to the tissue culture. So egg inoculation is done. Now coming to the tissue culture. So in tissue culture, the viruses are cultured in tissues. Now that those tissues may be of three types where we culture the viruses. That may be organ culture, that may be a explant culture, or maybe a cell line culture. Now among this, the cell line culture is most important and asked in the exams. Now for the organ culture and explant culture, just remembering the examples uh, will do. So for organ culture, the tracheal ring culture is used for the coronavirus. Coronavirus is cultured in the tracheal ring. For the explant cultures, the adenoviruses are cultured in the adenoid explants and these cell line cultures are the point on which we want to focus more, because these are most commonly used in modern research of viruses. So for the cell line cultures, we have to first prepare the cell lines. So coming to the preparation step of the cell lines, how do we prepare the cell lines? So for that, we take a tissue, whatever tissue we want to take, over which we want to grow the viruses, we take that tissue and we will be completely digesting the tissue so that all the cells get separated from each other, like they are cells are bind with each other by the extracellular matrices which are made of collagen. So we use collagenase enzyme and some other proteolytic enzymes like elastases and so on. So by using those such types of enzyme, we will make the each cell free from each other, so that each cell become free. So now we after adding the enzymes, we will mechanically shake, and due to that mechanical shaking, the individual cells will get separated from each other. The extracellular matrix will be dissolved. Now all cells are free. So we want to get those cells only. So for that, what do? We will just wash the cells, we will count the cells, and we will suspend the cells in the viral growth medium. So we'll take a viral growth medium and the cells which are washed, and after counting, we suspend those cells into the viral growth medium. And this counting and the washing is very important, because we want to remove those digested extracellular matrix material, and we want to get just the cells only. So this washing process is important. Counting is also important, because we want to see the growth of the viruses in the cells, and then we will suspend it in the growth medium. After suspending in the growth medium, the viral growth medium containing the cells are dispensed in the tissue culture flask. So we'll take a tissue culture flask and we'll keep that viral growth medium with aided or suspended cells into a tissue culture flask, and in and that tissue culture flask with the virus viral growth medium is incubated so that the cells just settle down and form a monolayer sheet at the bottom of the flask tissue flask. So that forms a monolayer sheet. That incubation forms the leads to formation of a monolayer sheet of cells at the bottom of the flask. And on that monolayer sheet, we will see the growth of the viruses, how the viruses are growth, and what are the characteristics of the growth of any particular virus. So now there are different types of tissue culture based on the type of cells we are using. So that first cell line is the primary cell line. Okay, that is the primary cell line. Primary cell lines are derived from the normal cells. Not here. They are derived from the normal cells. So here normal tissue is used for making that cell line, and these normal cells have the capacity to divide up to 5 to 10 divisions, and they maintain the diploid karyosome. An example for that is the monkey kidney cell line, and the chick embryo cell line, and the chick embryo cell line. Okay, monkey kidney cell line and chick embryo cell line. Now coming to the next cell line, that is the secondary cell line. Coming to secondary cell line, these are also derived from the normal cells, but they have the capacity to divide up to 10 to 50 divisions, and they also maintain the diploid karyosome. Okay, they also maintain the diploid karyosome. And for example of the secondary cell lines, we have human fibroblast cell line, the human fibroblast cell line, which is used for CMB, MRC5 and WI38. These two are the secondary cell lines. There are many more secondary cell lines also, but we want to focus on only few that is more frequently used, that is the human fibroblast cell line and the MRC5 and WI38 are the secondary cell lines. Next comes the continuous cell line. So these continuous cell lines, the meaning is that the cells continue to grow, they will not die, they will divide for infinite number of times. So basically, what type of cells can grow for infinite number of times? That means they are the cancerous cells. So they are derived from the cancer tissues. So they are cancerous cells and they have the capacity to divide for infinite number of times. And we know that in the cancerous cells, the diploid chiasm is not always maintained. Sometimes it becomes haploid and sometimes the ploidy changes. So the diploid chiasm is not maintained, and such type of cell lines, the example of such type of cell lines are many, and these are most commonly used also. So some of those continuous cell lines are the HeLa cell line, Hep2 cell line, McCoy cell line, which is used for chlamydia, Vero cell line, and the BHK cell line, So these cell lines must be remembered, as these are important cell lines and these are facts only. So one must have to remember these only. Okay, now once the virus has been grown over that cell culture, over the cell lines, so how will we detect whether the virus has grown or not? So there are different methods to detect the growth of virus in the cell culture. So detection of the viral growth in the cell cultures. So the first thing by which we can detect is the cytopathic effect. Cytopathic effect is the nothing but the morphological changes that is occurring to those cell lines due to growth of that virus. Okay, so cytopathic effect means cytopathy. Cyto means cell, pathy means pathy to cell. I mean cytopathy means pathology to cells. Okay, so pathology to cells means what are the changes occurring to those cell lines after the growth of the virus, that is the cytopathic effect. So definition is very easy, by the name we can say it easily. So there are certain cytopathic effects which we should remember. These are the particular cytopathic effects which are seen in these only in particular viral infections. So these cytopathic effects lead to initial derivation of the viruses which may have been inoculated into that cell line. So for example, if we see that there is degeneration of the entire cell sheet, crenation and degeneration of the entire cell sheet, then we can pre-assume that the virus may be the enterovirus. The virus may be the enterovirus. If we see multinucleated giant cells, then we can infer that it may be a measles virus or herpes simplex virus. So that's the importance of these cytopathic effects. By that cytopathic effects, we can pre-assume the viral infection. We can get a knowledge about the virus which has been infected. Similarly, the ballooning of the cell lines is seen in case of HSV, herpes simplex virus. Cytoplasmic vacuolation means vacuoles in the cytoplasm of those cell sheets are seen in case of simian vacuola virus 40. I think that is the name. SV40 is the short form. Then clumps like bunches of grapes is seen in case of the adenovirus. Is seen in case of the clumps like bunches of grapes in case of the adenovirus. So now the next way of detecting the growth of the virus on the cell lines is the viral interference. Is the viral interference. What does that viral interference mean? So viral interference simply means that if a cell line has already been infected with a virus, then a second virus cannot grow over that cell line. Okay, that is called as viral interference. Simply, the first virus which has been grown on that cell line is interfering with the growth of the second virus which we are adding. That is the viral interference, and that is in the definition as well. So what is the advantage of that? The advantage is, see here with the example, we will understand what is the importance of that viral interference. So for example, we have, if we grow the Rubella virus on the cell line, and Rubella is known to produce no cytopathic effect, so it will cause no cytopathic effect, no morphological change on the cell lines over which we are growing it. So if when there is no cytopathic effects, then we may think that there is no growth of any virus, there is no virus. So we will induce or we will inoculate a virus which is known to produce the cytopathic effect. And when we inoculate a virus which is known to produce the cytopathic effect, and if the cytopathic effect of that added or inoculated virus does not appear, then we can safely say that there has already been growth of some kind of virus. So some virus has already been grown over there. That's why the second virus which is known to produce the cytopathic effect is unable to grow there. So that is the importance of the viral interference. We cannot say which virus is that, but we can surely say that there has already been growth of some of the virus. That is the importance of the viral interference, and that is the definition as well. Now coming to the third point by which we can detect is heme adsorption, and this is important only in case of the heme agglutinating viruses. For example, the influenza virus is a heme agglutinin virus. Why? Because it expresses the heme agglutinin antigen. What is the function of that heme agglutinin antigen? So as it expresses the heme agglutinin antigen, so when we add the erythrocytes to it, it will agglutinate those erythrocytes. Okay, so that's why it is called as heme agglutinin antigen, and that is a function of that heme agglutinating viruses like influenza. So how do we detect those viruses? When these viruses grow over the cell lines, they will produce the heme agglutinin antigen over those cell line, over the cells of that cell lines. So those cells will be producing the heme will be expressing the heme agglutinin antigen, and when we will add the erythrocytes from outside, the erythrocytes will get adsorbed on the cell surfaces of those cell lines. That is called as heme adsorption. So by that method we can detect the heme agglutinating viruses. Next comes the immunofluorescence test. So by immunofluorescence test we can detect the virus growth. How? So to detect by immunofluorescence, we will take the virus infected cells and will make a smear over a slide, and we will stain it with antibodies which are tagged with a fluorescent dye. So when those specific antibodies will bind to their antigens which are expressed on those cell lines, then they will produce the fluorescence, and that fluorescence will be detected, and by that we can detect the growth of the virus. This is done under fluorescent microscope because we have to detect the fluorescence. So this is another under the fluorescent microscope. Next is the electron microscope. By electron microscope, the changes in the cell lines or the very minute changes can be detected. So that may be a method by which we can detect the cell lines, growth of the viruses over the cell lines. And the last but not the least is the viral gene detection. So by using PCR or other molecular methods, we can detect the specific viral genes, and this is the most most specific method to detect the viral growth, by which we can detect the type of virus or which virus is growing over those cell lines. We can detect it by these molecular methods. So these are some of the methods by which we can detect the growth of the viruses on the cell lines. This is all about the isolation of the viruses and detection of the growth of the viruses over the cell lines. Next, we will go into the virology part. The general virology contains this much, although contains some more topics, but this much is important for the exams. So we will be limiting ourselves to this much only in general virology. Okay.